This is part 2 for Chapter 16, Innate Immunity. As I explained in part 1, your immune system is defending you against infectious disease and cancer, and the overall organization of your immune system is as follows. Your innate immune response, also known as your nonspecific immune response, is comprised of the first line of defense and the second line of defense. All of this is protecting you from microbes in general. Your third line of defense is the same as your adaptive immune response, also known as your specific immune response, and this is where your immune system is responding to a microbe that is infecting you at that time. Last time, I went over the whole first line of defense, different types of barriers, different washing processes, chemical factors, and your normal microbiota. And again, the purpose of the first line of defense is to prevent infection, to keep the microbes out of your body. Then after I went through that, I introduced the different components of the immune system. So lymphoid organs, lymphatic vessels, lymph nodes, and the white blood cells to get ready to talk about the second line of defense. So this part of the lecture is all about the second line of defense. So again, second line of defense, this comes into play once the microbes have gotten past your first line of defense and they are infecting your body. They have gained access to your tissues. And this will involve a lot of the different white blood cells, leukocytes, that I talked about before, and it will involve different processes such as inflammation and fever, and then antimicrobial substances like complement and interferon. Just as a review of the different types of leukocytes, I went through them. So here they are in a different order. These are your granulocytes. Granulocytes have granules in the cytoplasm, so you can see those granules, and they are the eosinophils, basophils, and neutrophils. And these are associated mainly with your second line of defense. The A granulocytes are over here. These are the A granulocytes. A granulocytes because they have very few granules in their cytoplasm, and the two types are the lymphocytes and monocytes. Lymphocytes, again, these are the white blood cells you find in the lymph nodes, and they are part of your third line of defense, so I'm not really going to talk about them now. Monocytes, on the other hand, are immature macrophages, and the macrophages are involved in your second line of defense. The first process that I want to go over is phagocytosis. And phagocytosis, of course, is cell eating. So cell eating, phago means to eat, cytosis, cells, and this is the process by which some of your white blood cells, the neutrophils and the macrophages, can phagocytose microbes that are entering your body. And the obvious benefit is that phagocytosis ends up killing and eliminating the microbes. For phagocytosis, you are going to have to know all the different stages of phagocytosis, and this image shows you them in order. And for this image, this big cell is the phagocytic white blood cell, either a macrophage or a neutrophil, and this is the microbe that it will be ingesting, so here is the microbe here. To start this process, the white blood cell needs to be attracted to the site of infection. So that would be chemotaxis, when cells move in response to a molecule. So the very first thing is that the white blood cell needs to go to the area where the microbe is. Once it is there, then it needs to be able to adhere to the microbe, has to be able to attach to the microbe. Once the white blood cell has moved to where the microbe is and attached to the microbe, then the white blood cell will ingest the microbe. So it will perform the phagocytosis moving the membrane around to surround the microbe. And this will result in a vesicle forming called a phagosome. So this is the phagosome there. Once the phagosome has been formed, then the next stage will be digestion. In order to have digestion, you need to fuse the phagosome to a lysosome. And remember, lysosomes are vesicles that contain digestive enzymes. So in order to have digestion take place, the lysosome and phagosome need to fuse together, and the resulting vesicle will be called a phagolysosome. And now those digestive enzymes come in contact with the microbe and start digesting the microbe. And this part of the image shows the microbe being broken down by those digestive enzymes. Once digestion has occurred, then there will always be a few parts of the microbe that are indigestible, that cannot be broken down. And those parts of the microbe that cannot be digested will remain in the vesicle, and it will be called a residual body. And the very last stage is that indigestible material. So this is the process of phagocytosis. And it seems very similar to how humans eat, because if we want to eat something, often we have to move to where the food is. We have to adhere to the food. We have to be able to pick up the food with our hands. We ingest the food by taking it into our body. We break it down and then digest the food. And then not all of the food can be absorbed by our bodies. So we have indigestible material that is then discharged. So it's the same basic idea, just on a cellular level. The second process I want to go over is inflammation. And inflammation is technically a response to tissue damage, whether or not there is actually an infection. If you pull a muscle, you will have an inflammatory response, but there would not be an infection. If you cut your skin, you will have an inflammatory response, and there is also a high likelihood there will be an infection, that microbes will be entering through the cut. So if there is actually an infection, inflammation will provide 3 benefits. The first benefit is that it will confine the microbes to a localized area. Second benefit is that it will destroy the microbes that have entered into the tissues. And the third benefit is that it will then facilitate tissue repair, which will repair the breach to your barriers, your first line of defense. So again, inflammation is triggered by tissue damage. In this case, you have a thorn that is damaging the skin on a person's finger, which will trigger an inflammatory response. The 4 signs of inflammation. First sign is rubor, and this is Latin, but rubor means red. So in that area where you're getting an inflammatory response, it will turn red. The second sign of inflammation is calor. Calor is Latin for heat. So that area will turn red and it will become warm. The next sign of inflammation is tumor. Tumor means swelling. So that area that is inflamed will begin to swell. And the last sign is dolor, which is pain. So the 4 signs of inflammation, if you are having an inflammatory response, is that the area will turn red, it will be warmer than the other areas of your body, it will swell, and you may have pain. This slide will go through the process of inflammation in more detail, and it will talk about the white blood cells that are involved and also how the whole process works. So the very first thing that happens is injury. So that's injury to the skin, and in this case, a pin or a needle is breaching the barrier of the skin, And all these little green things are microbes that are on that pin or needle. So it was not a sterile needle. When the injury happens, that is going to stimulate the cells that are damaged, so the epithelial cells that are damaged, and mast cells. Mast cells are a type of white blood cell. Both of those cells are going to release chemical mediators, signal molecules like hormones. And the major chemical mediator that is released is histamine. So histamine is going to be what triggers the inflammatory reaction. Once the histamine is released, that will have an effect on the blood vessels that are in the area. And the two effects are that they are going to dilate, you're going to have vasodilation, and they are going to become more permeable. More permeable means that the plasma is going to be able to seep out of the blood vessels. So vasodilation, increased permeability, means that you are going to get more blood to the area and the plasma is going to be able to leak out, and that is that yellow stuff that is leaking out. So histamine release is going to be triggered by tissue damage, and it's going to stimulate the blood vessels to dilate, to bring more blood to the area and to become more leaky, increase the permeability so the plasma can leak out. This diagram shows in a little bit more detail what is happening during the first stages of inflammation. So down here somewhere, damage has been done. Maybe microbes have entered the tissues, and the damaged cells or other cells are releasing histamine. So that's the chemotactic factor. So these blue molecules would be those cytokines, probably histamine, that have been released by those damaged cells. So that histamine is going to trigger this blood vessel to dilate, to bring more blood, which includes more white blood cells and more antibodies and more complement to the area. And then the histamine is also going to stimulate the blood vessel to become more permeable, to become leaky. So then those white blood cells can leave the blood vessel and go to the site of infection. And of course, the first white blood cell that will be there are the neutrophils, because the neutrophils make up the highest percentage of white blood cells in the blood. You have a lot of them, and they will charge into the site of infection, and they're attracted to that area of infection by the chemotactic factors, the histamine. All right, so you had the damage was done, microbes have entered into the tissues, the damaged cells and the basophils have released histamine. That's made the blood vessel dilate, made it leaky, so that the neutrophils can leave the circulation and go start fighting the infection. So as this progresses, what you will have is a lot of neutrophils arriving to the scene to ingest the microbes that have entered through that damaged part of the body. And you will also start having pus formation. So pus formation, the pus is basically composed of plasma, the liquid part of the blood that has leaked out of the blood vessels, the neutrophils, and lots of dead neutrophils, because remember, once they ingest one microbe, then they die, and also it will contain microbes. So pus usually contains plasma, neutrophils, lots of dead neutrophils, and the microbes. And of course, during this whole process, you're getting blood clotting, so blood clotting, scab formation, and also that serves to isolate the microbe so it remains localized in this area, confining the microbe to the area. Then at later stages of inflammation, what you have are macrophages moving in. So at the very end of the inflammatory process, you have the macrophages coming in, and the macrophages big eaters, they are going to come in and clean everything up. So they are going to ingest the dead neutrophils, and they are going to ingest the remaining microbes. And they also ingest the plasma. So they basically come in and ingest, phagocytose all of the pus and everything that's there. Now the macrophages, it takes them a relatively long time to come in because these are wandering macrophages. And wandering macrophages are found only in the tissues. So macrophages are never found in the blood, they are only in the tissues. So it takes them a long time to get there because they have to crawl through the dense tissue to get there. Neutrophils get there very quickly because remember they're in the circulation, so they are circulating throughout the body, they're on the highway system of the body, so they can get anywhere very quickly. But the macrophages, it's going to take them a lot longer to get there. The third process I want to talk about is fever, and you should know fever is an elevated body temperature. Fever is actually a response to infection. So the fever response is much different than an inflammatory response. Inflammatory response is a response to tissue damage. Fever is actually a response to infection. And the benefits of having the fever are as follows. First, it's going to inhibit the microbes, and it inhibits microbes in two ways. It's going to inhibit the microbes' ability to replicate, and that would be both cellular microbes, prokaryotes, eukaryotes, and also viruses. Second benefit is it's going to inhibit the microbes' ability to take up nutrients, and that mainly affects the bacteria. It would not affect viruses, of course, because viruses don't metabolize. The next benefit is that a fever, an elevated body temperature, will actually stimulate your leukocytes. So it actually stimulates the leukocytes, makes them more active, and inhibits the microbes, which is what you want when you have an infection. You want to inhibit the microbes from functioning, and you want to stimulate your white blood cells so they can fight the infection. So now the process of fever, and for fever, the main cell that is involved is a macrophage. So macrophages are the white blood cells that are going to stimulate or trigger a fever. And what basically happens is that the macrophage is going to phagocytose a microbe. So here is your macrophage, it's phagocytosing a microbe. And when that macrophage recognizes molecules on the microbe, such as endotoxins. Remember endotoxins, basically lipid A, that will stimulate the macrophage to produce cytokines. So that will stimulate the macrophage to produce cytokines, and cytokines are molecular messengers between white blood cells. And the main cytokine it's going to produce is IL-1. And IL-1 stands for interleukin. Interleukin. Interleukin, inter between, leuko, white blood cells. So IL-1, and there are many ILs, IL-1, IL-2, IL-3, and so on. These are molecules that white blood cells use to communicate with each other. So once the macrophage endocytoses a microbe, it's going to trigger the production of cytokines by that macrophage. Those cytokines are going to diffuse into the circulatory system, and they are going to diffuse all the way to the hypothalamus of the brain, and they are going to stimulate the hypothalamus to raise the body's temperature. So this is the process of fever. It is triggered by a macrophage that has ingested a microbe. That macrophage secretes IL-1. The IL-1 diffuses through the blood vessel until it gets to the hypothalamus of the brain, where it stimulates the hypothalamus to increase body temperature. Complement. complement is a really, really important part of your second line of defense, but probably you have never heard of complement before. So I want to take a little bit of time to go through complement. Complement is basically a group of serum proteins. And so complement, the word itself, is a plural because it's referring to many different types of serum proteins. There are about 20 different serum proteins. And serum, of course, means the blood. So complement is found in the plasma, the liquid part of the blood, the serum. And complement is activated in a cascade manner. That means if you activate just one serum protein, that one protein will activate another protein, usually more than one, and each of those will activate more proteins. So that is your cascade manner of activation. And to activate complement, there are 3 different pathways, which I will go over in a minute. And once you have activated complement, there will be 3 results that will benefit and help you fight an infection. Now I will go through the 3 different pathways of complement activation. And the first pathway is called the classical pathway of complement activation. So in order for this to occur, you have the microbe here. This is the microbe, and you have certain molecules on the surface of the microbe called antigens. And for the classical pathway, you need 2 antibodies bound to the microbe. When you have antibodies bound to the microbe, and antibodies always have this Y structure, and they're always drawn in green. When you have 2 antibodies bound to the microbe, then that allows the first complement protein, C1, to bind to those antibodies. And cascade manner of activation, if C1 is activated this way, it will activate C2 and C4, which will then split and lead to activation of other complement proteins. You do not have to know the different numbers and orders of the complement proteins, but what you have to know for the classical pathway of activation is that it requires a microbe and antibodies. So having the antibodies bound to the microbe, that will activate the first complement protein. The second way to activate complement is called the alternative pathway. And for the alternative pathway, you just need the microbe. So only the microbe will activate the complement. And in this pathway, 3 different complement proteins called B, D, and P, you don't have to know what they stand for, they will bind directly to the microbe, and then that will lead to activating a complement protein, which will lead to it splitting and activating other complement proteins. The last way to activate complement is by the lectin pathway. And lectin is the molecule that you find in blood. And so for this activation, all you need is the lectin in the blood to bind to the microbe, and that will activate the complement proteins. So as long as you have lectin binding to the microbe, that will activate some of the complement proteins, which again, in a cascade way, will activate other complement proteins. So again, you do not have to know the order of the different complement proteins. What you need to know is that the lectin pathway involves lectin binding to the microbe to activate the complement. So lectin pathway, lectin binds to the microbe. Classical pathway, antibodies bind to the microbe. And alternative pathway, the complement proteins directly recognize and bind to the microbe. Once complement is activated, it's going to lead to 3 benefits. So these are the outcomes of complement activation. So the 3 benefits that will help fight infection. The first benefit is opsonization. And opsonization is an important word for the immune system. Opsonization is basically defined as having proteins coat a microbe. So all these little red things are the proteins, in this case the complement proteins, coating a microbe. So this brown thing is the microbe. And once you have all these proteins coating the microbe, that makes it much easier for phagocytosis to occur. So having a microbe covered in proteins will make it easier for the phagocytic cells, the macrophages, the neutrophils, to phagocytose the microbe. And in this case, having complement proteins bind all over the microbe will make it much easier for the phagocytic white blood cells to adhere to the microbe and phagocytose the microbe. Second benefit. The second benefit is triggering inflammation. So here's inflammation. Once one of the complement molecules is activated, that will lead to activation of other complement proteins, which will stimulate mast cells and basophils to release histamine, which will trigger an inflammatory reaction. So that is a second benefit of complement activation. The third benefit of complement activation is cytolysis, lysis of the microbes. And this basically occurs by MAC formation. So MAC, M-A-C, this stands for membrane attack complex. And this is the membrane attack complex here. It is composed of complement proteins. And what these activated complement proteins do is they wedge into the microbe cell membrane and they create pores in the membrane. They can create hundreds of pores in the microbe's membrane, and all the cytosol leaks out. And of course, that leads to lysis of the microbe and death of the microbe. So that's called MAC formation, membrane attack complex, and that's done by the activated complement proteins. So the three benefits of complement activation can be abbreviated by OIL, O-I-L. So O-I-L, O stands for opsonization, I stands for triggering inflammation, and L stands for lysis by MAC formation. The last thing I'm going to talk about in this lecture are interferons. Interferons are a type of cytokine. So again, cytokine, those molecular messengers, they send signals. Again, another way to refer to cytokines are interleukins. So interferons can send signals to white blood cells or to other cells. So interferons, they are signal molecules. And interferons are only antiviral. So they're not effective against bacterial infections or eukaryotic microbe infections. They are just specifically against viral infections. But they are still part of your non-specific immune response because they're effective against all viruses, not just one particular type. And the purpose of the interferons is to protect uninfected cells. So that way it can limit a viral infection. It can prevent uninfected cells from becoming infected. And this will be a diagram of what happens or how interferons work. So here you have the virus, and the virus is going to infect a cell. So you are going to have an initial cell infected. And that virus is going to start going through its life cycle. Remember the life cycle of viruses. You have attachment, penetration, biosynthesis, maturation, and release. So this host cell has been infected by the virus. Being infected by the virus is going to trigger this host cell to produce interferons. So there are two types, alpha and beta. You can just refer to them both as interferons. But being infected by a virus is going to trigger the production of interferons. And not only is the cell going to produce the interferons, but the cell is going to secrete those interferons out. so that those interferons can go to the neighboring cell and stimulate that cell to produce antiviral proteins. So this signal molecule is going to stimulate this neighboring cell to produce antiviral proteins, so that when a virus particle that either buds off or is released by lysis from the host cell, when that virus particle attempts to infect this neighboring cell, that will be stopped. So antiviral proteins, it's going to stop the infection. So interferons are signal molecules that are produced by a virus-infected host cell to send the signal to the neighboring cell so that the neighboring cell can produce antiviral proteins. As a review for what I have covered in Chapter 16, we have gone over specifically the innate immune system, which is also called your nonspecific immune system, and it is composed of the first line of defense and the second line of defense. First line of defense is meant to prevent infection, and it involves how your body is structured, the physical factors, the types of tissue that cover the outside of your body, and their chemical factors, their washing processes, and your normal microbiota. If those microbes get past your first line of defense, then they have entered into your tissues, and now it is up to the classical parts of your immune system, the white blood cells, to defend you, and processes like inflammation, fever, and antimicrobial substances such as complement and interferons. The next chapter, Chapter 17, will be focused on your adaptive immune response, how your immune system adjusts to a microbe that is currently infecting you.